topic today is what's a gene, and it's a serious discussion because I don't know if I can conclude today's discussion without the help of Dr. Deal to give you the right answer. But I want to try and just tell you a little bit about maybe the history, the personal and molecular perspectives of a gene. And why am I doing that? Because there are 26 people in some place that will be asked that very question a week from this Friday. What's a gene? And that's a legitimate question to ask from a person who's a geneticist and from a course that they took called Biology 222, which says genetics. And so we're talking about genes. And yes, indeed, it's done by Dr. Gabe, a.k.a. Gregor. And final thoughts, of course. Well, look at this guy. You know he's my hero, and I know that many of you have adopted him as, in fact, a person to whom you look up to daily as you say your prayers in the morning, thanking God for those P's you've been given. But in fact, I got some sad news for you. Gregor's here pensive, if you will. Can I use the word pensive in this class? Very good. And he's got his Bible out, praying for a little help. Did you know that? Did you know that our father of genetics never used the word gene? The gene word wasn't coined until after 1900. This poor gentleman went to his eternal rest in 1884. So if we're going to talk about genes, Gregor's sort of off the hook, but I don't want you to be disappointed because many of you memorized a lot of stuff, but maybe he could have used the word factor. All right, so he didn't use the word gene. The story's still okay. Are you with me? By the way, the answer is amen, which I think is Aramaic, which means so be it, I think. Well, let's practice that a little bit. Are you with me? Amen. Very good. So if I put these pictures up, you can be very comfortable with saying, oh, yeah, I remember that. We've got the tall and the vertically challenged. We've got the purple and the white. We've got the full seeds and little seeds and all those crazy things he talked about. And as a consequence, you would have come away with the idea that a gene produces some crazy trait. Now, is that a correct kind of start? Well, okay, if your name is Gregor Mendel, fine. That's a great start, I think. Is it still relevant today? Yeah, sure it would be. But we've got to go beyond this because we've got about 30 slides to pump through to figure out where does Gabe want to take me with exploring the idea about a gene and what, in fact, it ought to mean. And we'll get there sooner or later. Now, if this is you, and you're on the front, not on the back, the night you were conceived, okay, maybe you didn't get the full deck. I can't help you. But in fact, the guy in the back has probably been doing steroids of some sort, and therefore he's augmented his genetic stuff with an environmental boost. So if you're psychology here, and there must be someone from psychology, you would certainly be happy with the idea of nature versus nurture. And okay, maybe you ought to put a tattoo on too because you could use some help. Well, for the people who are actually, 26 people in this class, you take out your book because you paid a lot of money for it, or maybe you're renting it, but you've already sold it because you need drug money, and so you might not know in the glossary. It has a good definition, which I want to read to you because I want to develop the idea that perhaps the concept of gene is a little bit squeezy or a little bit something. It's just not quite there. The fundamental unit of heredity whose existence is confirmed by allelic variants and which occupies a specific chromosome locus, a DNA sequence coding for a single polypeptide. All those who like that definition, please raise your right hand. Those who thought that definition was lacking, please raise your right hand. Well, that's my left hand. But I had to raise the same one that would agree with you as you're sitting there. All right, I agree with you. That definition leaves something to be desired. In a different setting, I might use different words. But this is a setting in which I say leaves something to be desired. So let's go on to see if we can try to see what might be an appropriate answer for what's a gene. Well, I teach a course at 8 o'clock called Medical Genetics. A book published in 2015 tells me it's a fundamental DNA that is used to make valuable products. If I would have written that, I would be so embarrassed to stand in front of you to say a valuable product. Now, if you studied Bayer, you might like scat. But if you know what scat is, it's not too valuable, except to a scat specialist. And so as a consequence, you know, that doesn't sound too good. And a factor controls phenotype, who cares? Again, I'm left short, and it's never good to be left short. Left short in the sense that, hey, this doesn't quite make me happy, if you will. So I go on. Oh, I know what I'm doing. That's good. And by the way, that's Emma. That's 1/8 of me right there. Could you have applause for Emma, please? All right, there you go. There are several other eighths in the audience, and we'll talk about that at the very end. If you don't know your math, don't worry about it. Michael will explain it to you. Whoa. Because I went to two places that I thought were quasi-authoritative, and because I sat down and put my hand on the Bible and asked Gregor to guide me, I want to know what a gene is. So I'm going to the source in whom I'll have tremendous confidence, our government. Now, you should have known that because you see the gov there, and so National Library of Medicine, National Institute of Health, and you might not know what that means. It's called genetic home reference. It's sort of cute. Everyone should have a genetic home reference. It's a big, long statement. I'm going to read you fast, and our game is to try to figure out what a gene. We got 30 minutes to do it in, and if I fail and you can't do the test and complete the test online and get a good score, obviously, I'll probably have to hit a little monk wine later this afternoon. A gene is a basic physical unit of inheritance. I guess gene is passed down from parents, containing information needed to specify traits. Hey, the government's telling me about birds and bees. I think they're really pretty good at that birds and bees story. That's nice. Genes are arranged one after another in structures called chromosomes. It contains a single long molecule, and they get some genes on it. Gregor would have appreciated that. I'll tell you very well, because he might have used this for an explanation. In fact, the video that you've seen, if you've taken genetics, he sits in the woods and contemplates genes and said, oh, it's like beads on a string. And so I brought his beads with you, straight from Bavaria, to, in fact, use it to try to tell you the government's idea is okay. It's not okay. That's a horrible definition. And then it down here tells me he had 23,000 genes. That absolutely, government failed me again. Those who didn't see what that was, why they're giggling, those who didn't see that, are you sleeping on me already? You need an afternoon nap? Well, at any rate, it stinks. So I want to quiz my class because I'm proud of what they've done this semester. And they'll make me equally proud if I can get through these 4 questions and not falter. By the way, anyone may chime in because I won't know you're not my class, and that'll help the people here thinking they're going to be picked upon. Simple question, class. Yell it out with pride. How many chromosomes does a human have? Oh, that's a lot of pride there. All right, class, how many chromosomes does a human have?
46.
Very good. We're 1 out of 4 so far. Class, how many base pairs of information is found in the genome of a human? You know what this is called? Mumble, mumble. All right, so I'll give you a hint. Well, hold on, you knew that. You all should have known that. But that as you've taken genetics, you ought to realize there's 3.2 billion base pairs. Now where are we going now? Oh, the government tells me there are 23,000 genes, but I don't think so. Class, how many genes are in a human? Let me pick some numbers out of my class. Let me think the number that was good. Let me see if his face is here first before I call his number. By the way, we each have a marble with a number on it, and alphabetically they have been given. I pull in marble every once in a while and ask people questions. The most exciting question was asked for Dr. Jeff, of question for number 14, man. I don't see 14 here. Oh, number 14 is sitting right over there. So we'll cut him a break and we'll push a button. There are up to 21,000 protein coding genes. And then the last fact I want to do is something that you might not be totally aware of, and we can't go there too much, but someone once said there are 19,000 pseudogenes. Now, because of the liberal arts education you've got, you ought to understand what the word pseudo means. You might not totally understand why there are 19,000 pseudogenes. I might not understand that either. But a long time ago, 2006, somebody came up with a statement. These are broken fragments, maybe even pretty good continuations of looking like genes, but they're not quite genes. Something's wrong with them. They can't be fully expressed. Holy mackerel, I got 19,000 things that don't quite work, but I'm going to be careful, and I'm going to copy them faithfully because maybe someday I'll need them, I don't know. But this is really the important stuff that we're going to look at. So what the heck does a gene do? Well, to do genes, we would have to know about DNA, so I've got some DNA here, and because we're not in the new science building, I want to know that DNA, in fact, is quite an exciting molecule. It vibrates a lot. we get to the new building, we can have the vibrations occur and your seats can shake, but that's not yet. I've got to go back and shake the slide just to show you that DNA is going to be found in a watery environment, and it's an exciting molecule. It's boring, people. A, T, C, and G. A, T, C, and G. That's it. And you can see them up there in the center, and I've got to at least review with you all, just in case you're an accounting major. Time out, please. All the accounting majors, please raise your hand. Do we have any accounting majors there? Oh, good. I'm going to talk to you because I got to make sure you understand DNA. All those people who love accounting majors, please raise your hand. All those who just came through tax season and realized that person just saved your, that's good. We go on. Our goal then is to see what the heck this gene, oh no, it's time for a money question. Now, they've been promised when the cat comes up, it's money time. Now, if Mason's back there, Mason would be running up here for the money. And I don't know if I can get to my, my wallet. Excuse me, just, I hope you didn't see the boxer shorts. They got pea plants on them. Oh my gosh, not good. All right, we got the wallet. I have a simple question for someone. You will stand up. You will say who you are. You'll say where you're from. You'll answer the question. If you're wrong, you will leave the room. Hey, this is life, people. Your college existence is a bit pampered. Let's talk about reality. The question is very simple. I'm going to explain it very clearly. I'm going to actually choose a person from the back of the room I just saw smiling, and she's in the 8 o'clock class. When I say her name, she is going to try to hide. Do you know who I'm talking to back there? Did you see that my eye caught your eye? We had breakfast together this morning, the entire class. Katie, it's you. Katie, would you stand up and take 2 people with you? Is Weave there? Weave, would you stand up, please? Berger, stand up, please. Quickly. The 3 of you can answer this question. All right, these are 3 individuals who are in my morning class. They don't know they're going to be called on, but if they're going to be wrong, the class will vote in the Roman style. Survive or die. The question is this. I have a cat. By the way, this is my daughter Kristen's cat. So wherever she is over there, this cat's been with us for a bit. And the cat loves to... What? What is it? What did they do? I didn't hear that. What you said? Don't worry about the cat. But this morning they told me I can't feed the cat bacon because it'll kill it. I'm not going to respond what I was thinking of doing. All right, we go on. Your question is quite simple, and I've got to make sure we're on time. Your question is simple. It's a real question. It's not a facetious question. It's not a tricky question. It's not intended to embarrass you. But if you fail me, I will be embarrassed. I have your DNA, young lady, in the freezer upstairs. I know that, and you can shake your head. We have your DNA upstairs. Is that correct? We do. I don't know if that's legal, but we do have it up there. By the way, a lot of these slides are stolen, so if you're looking for copyright, maybe not. All right, so young lady, I want to know, relative to your, are you paying attention to me? Relative to your DNA, I get the DNA from a cat. I can do a cheek swab on the cat. The cat likes it. And so I'll get a cheek swab, get some cheek cells, extract DNA from the cat. In terms of biochemistry, in terms of the structure of DNA, how does your DNA compare to the cat DNA? Would you talk with your 2 people, please? This is like you have a lifeline. Quickly, we don't have a lot of time. This class has got to move on. Oh, by the way, how much money does she win if it's correct? How much? Do you have your answer, please? Why? And I guess we can go back that picture. So yeah, whether you're an orangutan or a cat or a dog or sheep, you get the same DNA in you. The thing that makes your difference is the sequence of those boring letters, A, T, C's, and G's. Did you get 5, 10, or 15? How about a $21 bill? What are we giving this young woman? Where's Mason? Mason, could you run down? Mason, come down here, please. Come on, let's go. Mason, quickly. You want to come down? Mason, you get money for the kids. Would you give this to her, please? And then you may have one of these. You like the money? You want 2 monies? Mason, you want 2 monies? Very good. We go on. So on to the idea that we know what that DNA is the stuff that genes are made of. You're welcome. And I want to recognize a really weird thing. And again, my government tells me only 1.2% of those 3.2 billion base pairs of DNA you have will actually make functional protein. All right, so that's an interesting kind of game, but that seems a bit extreme, but I think it's truthful. I'll let it sink in a little bit. Holy mackerel, that's crazy. That's really crazy. I want to move on with a different idea of what a gene might do. A guy named Beadle and Tatum, there were two people there, two guys named Beadle and Tatum, talked about the fact that maybe a gene makes enzymes, and so I want to recognize the fact that you've all been tested for PKU disease, phenylketonuria, whatever that may be, and in fact, it's relatively rare, thank God. It affects people because they don't have an enzyme, and then in fact, they need strict dietary restrictions for the rest of their life, or they will have trouble. Trouble means that they will, in fact, cause some of the neurons in the brain to, in fact, be destroyed. This is not good. So I'd like to show you something for the biochemistry people who are forced to be here. Biochemistry, raise your hand, please. The whole biochemistry class in here. All right, so here's your job for the weekend. Memorize this chart. Where's Dr.? Oh, there's Dr. McDonald back. And so this would be a typical chart they're memorizing, which is a real good activity for your brain. What we're looking at is there's an enzyme here. The red line must mean, well, I can't take this to go to this. But hold it. I have accounting majors in the class. So as they say in that language, only Pierre knows. Excusez-moi, s'il vous plaît. This is for us. All they're telling me is I have an amino acid, phenylalanine. I'm supposed to be able to make it into tyrosine. My chemistry says I'm going to, I forgot. Oh, it says it's hydroxylase. It must put a whole group on that crazy thing, on a phenylalanine, to make a tyrosine. For the rest of us, that's a hydroxide. If you don't have the enzyme because your gene wasn't working, you're going to have a disorder, which is unfortunate, but you will. And so I'm saying, okay, that's a new idea. Maybe a gene is responsible for making an enzyme. Is there legitimacy to that? Sure, why not? We got to go on quickly because with time is always an issue. In order that we could all leave this class and say, oh my gosh, I actually understood what a gene is and what it can do and what it's supposed to do. And by the way, obviously, if it fails you, you might have a problem. So I would just want to go back and talk about the central dogma of genetics. If there's a religion major in class, then that word dogma ought to, in fact, cause your heart to pitter-patter. But now where's my NGA? Because when I said that the other day, she wasn't quite sure what pitter-patter meant. So I explained it to her, and then she goes, yeah, you're right. My heart's not pitter-pattering either, because the thing we're talking about wasn't too exciting. This is exciting. They were saying, hey, look, you got some DNA? Yeah, we all do. And we're going to make some RNA, and then we're going to make some polypeptide. And they're just showing you a little tiny game or cartoon of where I got DNA. I'll make a piece of RNA that copies it. I'm going to put a tail on the RNA. I'm going to put a cap on it. I'm going to throw parts of it away, throw some away, and then squeeze it out a hole and bring it down here to Willy Wonka Chocolate Factory. We call those ribosomes. And I noticed they put the ribbon in the ribosome, and I got some spark plugs assembling little tiny purple things. Hold it, class. What are those little purple things that are being assembled? Quickly? Amino acids. So for those who went to Cockroach Cafe yesterday, you ate cockroach protein. Doesn't do you much good. But once you break down that protein, it's legitimate, whether it's rattlesnake or cockroach, that you can reassemble the amino acids in terms of what your instructions tell you to do to make the polypeptides you need. All right, so that's just the main machinery that I want to make sure we're all comfortable with so we can keep on the same level as I delve more into this question. What is a gene? Hold it, class. Do you understand what you just saw? Where is my class? Would you raise your hands, please? Number one, you shouldn't be scattered around. Number two, you missed your cue. Okay, there's a cue coming up. Are you ready? I know now why I'm retiring. This is not a bad diagram, but again, they always fall short. They always fall short. And so let me just go quickly through. There's a sequence of DNA, and they're telling me here's a place where I can regulate, transcription factors can jump on there. That means I can either make or not make RNA. I got a Tata box. That's interesting. Remember, the only letters in DNA are A, T, C's, and G's. And so a Tata box is interesting. For those old enough, that's a Don King kind of place. That's a promoter. And so I got a Don King kind of guy saying, hey, come on over here. let's read this gene, and then I got a start site. I got an exon. I'm throwing these away. I got that one. By the way, it's in green. Gregor would have loved the green. And that's why there's green everywhere here, because of Gregor. It looks hideous, but we're going to live with it. And then on the end, I'll put a tail on it. And once this is made into RNA, I would have then a gene. Woo. That is a woo experience. Now, I debated with myself, which I don't often do, how much this next question is worth. But when I tell you it's worth 50 bucks, Dr. Jeff is going to come up down here and say, I know the answer. He is ineligible, but I don't think he can find it either. All right, you ready? Have you thoroughly studied your gene? Have you thoroughly studied? Just shake your head. Are you thorough? All right, good. Find the gene. It's there. Can you? Oh, he's getting angsty. Oh, he's got to find. But hold it. So listen to me. No matter if you're an accounting major, a philosophy major, you're a double major, triple major, quadruple major, whatever you are, you already know how to find a gene. Oh, this one over here is pointing back and forth, and they're going for it. They want the 50 bucks. By the way, do I have 50 bucks I'd give you if you could tell me? I don't think you're going to find all the parts. It's there. Now listen, the computer will find it. Computer's going to find it real quickly. There it is. There is a one of those like control sites, of sense it's a binding for a protein. There's a start signal, and then these blue things are going to in fact be selected. These things will be eliminated. They're called introns. Now that seems a bit stupid. Is that a nice word? It seems a bit foolish that you would do such things. It'd be foolish even more to recognize you only use 1.2% of your DNA to code for functional protein. So let me just ask a question to my class. $5 question. Who wants a $5 question? Right here we have a young lady. This is Kaylin. Kaylin, would you stand, please? Would you say hello to the audience? $5 is the question. No, you're not getting more money. She wants money. No, you're not getting money. Oh, she can help. Oh, you may stand up, please. Would you say hello and where are you from? Because you're quasi-local. Well, may I help you? Come on. Let's just, you're not old. Stand over here, please. Would you say hello to the audience, please?
Hello.
Name?
My name's Alex.
Very good. Between the two of them, I guess I got to give them $5 a piece if they can get the right answer. I want to know, because I get a sense that some of these letters just aren't particularly important. What's the minimum number of letters? Are you listening to me? What's the minimum number of letters that are known, once they're changed, to cause a disease? Do you understand what I just said? I want to know how many letters can you, give me an example of a genetic disease where there's a change, and what's the minimum number I've got to change of the letters to cause a disease? Would you tell the class what the answer is, please? They say the answer is 1. You win $5.
Oh, thank you.
You win $5. By the way, these have been copied on the color machine. They look pretty good. All right, for the rest of you, you say, hey, I knew that because I know the disease. One example. There are lots of examples, but here's a disease, and when you see it, it's really devastating disorder. Are you ready for the devastating disorder? All right, so Dr. Fisher's going, as if he knew there was a one-letter change, but I think he did. There's a one-letter change that causes this disorder to occur. Those who know what the disorder is, raise your hand. There should be half. Okay, good, fine. I don't have to tell you then, but I have to tell you this. I've got to tell you the fact that, I've got to tell you a little bit about hemoglobin, and then I want to take you to the exact change. It is a one-letter change. So of the 3.2 billion base pairs that you've got, as little as one letter change can cause some serious harm. It depends what the change is, quite frankly. Let me quickly talk about hemoglobin because I want to show you the mistake is going to be in the beta chain. Go ahead. The mistake is in the beta chain. This is amino acid 1, 2, 3, 4, 5, 6, 6. I can't count. That is a glutamine in position 6. The letter causes and DNA causes a change to valine. That's a bad mistake. And I just want to tell you that the beta chains, there are 2 in your hemoglobin. They're a darker blue. And this is not a great diagram of it, but I like the little tiny disc they put in there, so I took this diagram. There are 2 chains, alpha in front, slightly less blue. So I'm telling you that there's a letter change in DNA, in a beta globin gene, 1 letter change, 1 amino acid change, and you got a person with a huge bad disorder called sickle cell anemia. And as a result, these crazy molecules, which are actually in the tens, hundreds of thousands in this red cell, So hemoglobin's small, sticking in this red cell, and then it starts to cause the molecules to adhere to each other, and it causes the darn cell to sickle. That's not good. So I guess that's an interesting idea. Genes must be important. Boy, but I just hope I don't change too many of those letters. Let me ask my class a question, please. All those in my class, please raise your hand so I can see you. Now, with the other hand, if you were a mutant, please raise your other hand. Very good. I've trained them well. They know they're all mutants in the sense that in your DNA, on average, there'll be between 1 and 2 million letter changes in you that might or might not be significant. Did I say that again? 1 to 2 million letters in you are different than the standard, whatever the standard is. And as a consequence, sometimes you get away with it, sometimes you don't. Holy mackerel, you missed another cue. Did you see that? Whoa, Mark, that was astounding. When I found this picture, I talked to the cat, no one else was up, so I'm talking to the cat, saying, cat, that's really a nice picture. Anyone else talk to cats? Okay, good. I don't want to be all by myself. I feel like Dr. Thayer, now retired. We're looking at the enzyme, a polymerase that replicates DNA in a bacteria. And I put it up here for the following reason. Number one, it's colorful. I like color. Number two, I don't even want to explain what it's doing, other than I know it's replicating DNA. But I go up here, and I would have, you know, Mason and Evan come up here and just count. We'll count 1, 2, 3, 4, 5, 6, 7, 8. I don't know where to stop. Here's a helicase. That's cute. There are all these polypeptides together that are required to make this work perfectly. Or hold on. So we started with Gregor saying that traits, genes called traits. Okay, Gregor, give you a check. Then we had a guy say genes can make enzymes. Okay, I'll give you a check. And then you have people say, well, genes make polypeptides. Give them a check. And then I look at this one functional product, and it's got all these extra parts in it that are required to make it work correctly. So what I'm doing is giving you a sophistication at a level of more of a molecular story that genes are got to work together to make all these parts to come together to make it functional, if you will. But I feel sort of rich today, so I want to give another opportunity to one of my students. Do I have a student who would like to volunteer? Well, thank you, Dan. Would you stand up and say hello to the audience? Oh, Dan, Sean, if you sit back in that back row, I'm not talking to you. Would you say who you are, where you're from, and wave to the class, please?
Hello, I'm Dan. I'm from Genomic.
That spontaneous applause is a recognition of the fact it's one of my favorite places. Who's going to help you, please? Quickly, we got to get this done, and we got to win money, and we're still doing okay on time. Kelly sits beside him in class, and so the two, would you come up, please, Kelly? Say hello to the class, and we've got a question for you. It's worth $5. Here someplace. Don't let me get in your way. Go. Are you ready? Oh, did you tell who you were?
Oh, yeah, I'm Kelly.
Where are you from?
Pittsburgh.
Thank you. Did you say hello to the class?
Yeah, hi.
Now, I'm going to ask the question. Don't yell the answer. Just like Price is Right. Don't be yelling answers out. I want to tell you a question that I see up here. And if you, biochemistry people, you should know this. I'm not sure that they're going to do this, and maybe I'll give them money if you vote for them to get money anyway. You know, because you got money, you might as well give money away because it doesn't do any good, particularly if you're dead. Here's my story. I noticed there's a word holoenzyme, and I was thinking holo, like, but that can't be. And so I tried to come up with a better idea of what a holoenzyme was. Biochemistry people, raise your one hand, please. You got to raise one hand up, biochemistry people. All those in biochemistry, leave your hand up. If you know the answer, raise your hand. The other hand. Then you have both hands up. Dr. Jeff, would you sit down, please? You're starting to make it. Yeah. All right, so I guess I really got a question that's too esoteric to be fair, I think. But I was amusing. By the way, let's shift them to another question since we got a minute or two. I noticed there's another thing over here. If you can yell this one out. I noticed they had something called SSB-coded DNA. And I thought for a minute, huh, interesting, yeah. You've all been told that, too, if you had genetics. You know what an SSB does. You don't, do you? Okay, I'm starting to have my heart cords ripped away from my body. I'll give you the answer to that, and then we're going to give them a guess, and then you're going to decide whether they deserve a special treat on this auspicious day. Tanner, that thing means single-stranded binding proteins. I told you when DNA opens up, the first thing it wants to do is go back together. So you put the proteins on it so it doesn't go back together until you have a chance to replicate it. Single-stranded binding proteins. Boy, this replication process is pretty complicated. Have you any idea what the heck a crazy holoenzyme is?
Well, I know DNA polymerase 3 helps hold everything together when it's replicated.
What's DNA polymerase 3 supposed to do?
It's supposed to help it.
Do what? What are you doing? What is this? Kelly, what is that enzyme doing? I just told you when I came up here. Oh, louder, please. All right, I gave her 2 5s, and she's got to decide if Dan earned any part of that 5. You know what I'm saying? What if I don't have 5s? I think I went to the bank and got a lot of 5s. My wife's going, well, Gabe, you went to where? Would you bow, please? All right, so I've taken you through a morass of weird things just to identify the fact that sometimes a gene can't do it by itself. Sometimes a gene might have a small mistake and cause a huge problem for you. So I thought this is absolutely an exquisitely beautiful diagram of a very complex but yet absolutely essential protein in the bacterial body. And we have the same thing too, different name. So I want to come to my glorious, it's not over yet, people, but I want to tell you about what I think a gene might be. I think a gene is a sequence of nitrogen bases in one strand of DNA that codes for a polypeptide, sometimes non-coding regulatory RNA, never talked about that, Gabe, and then sometimes maybe RNA for a pseudogene. Haven't talked a lot about that either. All I'm saying to you is that when I read my government report and they told me what a gene was, they never mentioned non-coding RNA, never mentioned pseudogenes. Why didn't they want to scare people? Because the reality is perhaps this gene thing is a little bit crazy in terms of what it actually can do. It says this gives you an opportunity to explain why we can utilize more of that 3.2 billion base pairs of genetic information that we so faithfully copy every time we make a cell. We copy all that thing whether we need it or not. And by the way, we copy all those pseudogenes. And sometimes, as in the case of this polymerase you've just seen, it might take several polypeptides or several genes to work in collaboration in order to make that product. And by the way, you better make the right number of each of those. Otherwise, you'd have a product that is not going to come together correctly, and you're still in trouble. What is that called? That's called genetic disease. That's sort of sad. Now, I know that you, in fact, are very excited about this, so I would suggest by your bedside you would have a book by, it's called Lewin's Genes 11. You can have my copy. You want to see what it says? 4. I've been trying to sell it to the book buyer for years. He just doesn't take it. I don't know why. In other words, that's a real nice resource. And did you see this? Whenever this was published, they just spent a whole book's worth talking about a gene. Holy macro! Yes, and you can have several graduate level courses, and you still wouldn't understand what a gene is. But my game is just to show you a little bit about it because it's an interesting topic. Well, Gregor says, forget about the genes. Let's do some P's. Class, are you prepared? Are you prepared? That's the question. It's not rhetorical. Because the old abbot says, oh, thank you, God. Our Gregor has given us peas. The dog's not particularly happy with it, although my dog would eat it. His fellow monks seem excited about eating peas. But Gregor, in true spirit, says, you're correct. And so, Gregor, you're quite the man, I guess. And so, Gregor went on to his eternal rewards. And mostly recognized that the work he did was not accepted to way after his demise. But we all have to have a demise someplace. 1884, they didn't know about his laws and know they were correct. It took into 1900 to do that. So there's my little educational historical fact. And that's sort of sad. But, all right, he was correct. And his things do work still today if you stay with the right kind of trait. I mean, you got to watch out. There's a whole bunch more known. So I consulted with Gregor, someplace up there in the blue. And Gregor said, oh, aren't genes wonderful? And then he had a second question. And I've been asked this several times in the last several days. What's next, Gabe? Well, you know what? I'm going to tell you right now. This June, my wife, who's back there someplace, and I will be married for 45 years. Can I have a little applause, please? We're on time, people. We've got to keep on time. And so I would tell you that I've been blessed, because Gregor would have used that kind of word, that we've had a very wonderful existence together. So I guess I must consult with my wife and decide what we're doing. I know what we're doing, but we've talked about it before. So you've all said, hey, what's happening, Gabe? As if there's nothing else to happen once I leave this cement floor and carpeting to go to other places. Well, other places might be nice. I don't think too much exotic thing will happen, but in fact, family is an important part of it. So it might be that I take a bike ride. I learned to ride bikes early. This is me, people, 8 months old. There was no metal at that time, and so this is a wooden tricycle. She actually believes it. She's believing that. She's true. But in fact, we had a fun ride one time, and Jim Spencer isn't here, but, and you can explain, what the heck are we doing here? Those are Lycoming water bottles in the old days. They were never orange until I spray painted them. So maybe I go biking. Maybe I go to the museum. People ask, could I put my picture in your PowerPoint? I said, yeah. So I put some oh my. Yeah. At any rate, I want to thank my 4 adult children. There's Scott sitting back someplace. My daughter Kristen. By the way, who's getting married this October 10th? My son Christopher. Mason was the one who's distributing money. My daughter Mary Beth. You see 1, 2, 3 grandchildren here now. That's wrong, because this was done 5 years ago, give or take a year. And so there are a few more kinds of things that go on, because I guess I'm going to be, and I want to thank Betty up there for my sign. Do you see the sign, Betty? And this is the current count. Now, genetically, we're not over with, people. I've got time. I've been hustling to get through this stuff. Genetically, then, I made the statement that the 1/8 of my genes is in those grandkids. Think about that in terms of biology. And it's a simple little mathematical game, because only half of my genes go to my son or my daughter, and then half of their genes goes on to their kids. And so start doing a little bit of multiplication theorem, and you'll come up with the idea that I have 1, 2, 3, 4, 5, 6, 7, 8. And today Mason's here, and so you can say hello to him. Olivia's here, Evan's here, and Emma's here too. She's here. Well, she was going out, and we congratulated her on being forceful and good, you know, because in fact it's good to breathe and take yourself into different places, perhaps. We're here at last summer. It's really fortunate to have them all together, so we'll see if that can happen again. But I can't be here unless I can have parents. So I hope that you appreciate your parents, and I want to just tell you a quick story about that, because this is my mother. I'm not sure what year this was, but my dad was still alive, which is nice. My mother lived to the lovely age of 93. Consequently, I would assume I've got some pretty good genes into me, which is good to know, because maybe I'll have a year or 2 left in me. So, by the way, if you want to, well, by the way, don't shake your head. We all got to look to the fact that there can't be an endless future. So I set this up. Look at outside, when you pick those cards up, when you become rich and famous, I'm not. When you become rich and famous, you're welcome to contribute to a scholarship fund I established on their name for students in biology or business, so that we can provide more of an opportunity, much like many of you have been given scholarships to be here. I've set one up for my parents. Why? They had 8th grade educations, and I'm very proud to tell you that, even though that's weird. But when you're old enough, you didn't need an education. You can work at the factory. You see what I'm saying? So that's no longer the case. So that's out there if you want to know. Where might I be? Might be at a beach. It's actually Scott and his wife and their dog, but it is the beach we'd go to. And we can sit on the beach. I like the beach, but I don't want to sit there too long. I don't want to be fried. So that's a possibility. And a number of people said, hey, can I be in this slide? I said, sure, there, you're in the slide. So Alicia, you're here. That's good. And this morning we had the Last Supper, sort of an idea. So the Last Supper was my 8 o'clock class down at Perkins. And we had a delightful time down there as we shared eggs together, I suppose. Oh, it's so dark. It says, thanks for coming. Any clouds? I didn't realize that was so dark. Thank you for being here. Other questions from you? Any questions from you at all? By the way, thank you for being here. This is a crazy thing to do, but I want to thank the president. I don't know if he's here. If he is here, this is a different thing we've established over time. And the fact is that this last presentation, last lecture thing is sort of a nice idea. And you want to come back in? Come on, let's go. Let's go. Any questions? Any questions? President something back. Thank you for having this kind of event possible. And we'll find out what they're going to do with that tape. I don't think they're going to be able to see these slides unless they've got a better system than I would think they have. So who knows what it might look like. Hold it, Dr. Mel, please stand and talk very loudly because you know I can't hear. I'm hearing impaired. They're going to have to know what a gene is for sure. Let's have, I hope there's a whole bunch of snacks, or maybe Gregor's got to go up there and bless them. If you need, Dr. Mary has a couple things to say, so I want to invite her to the dais.
First of all, I want to thank Dr. Gabe's family for sharing this wonderful man with us for all these years. Thank you so much. Let's give them a round of applause. We reluctantly returned him to your tender embraces, but not full time. I hear that HR gives retiring professors a certain set of gifts. I've heard that includes a rocking chair, a pen set, and a gift certificate to the bookstore. Now, you guys know Gabe. How long do you suppose he's going to sit in that rocking chair? Maybe 2 days. So we decided to outfit the rocking chair with some things he might need to get better use out of it.
Are you going to hold this so I can go like this? Hold it, here we go. Oh, Dr. Fisher would really like this one. Oh, there's one more thing down in there.
We have rocket boosters, and importantly,
Thank you very much. And seniors, I'm going to be with you also for graduation. I'm graduating with you. The rest of you having the finals, good luck on the finals. For those who came in, by the way, there are a number of people came in from all kinds of places. I don't even want to start talking about the faces I've seen that I haven't seen for a while. Thanks for being here. And indeed, I hope you understand a little bit more about what a gene is. Thank you. That's perfect.